Hi everyone. First of all, thanks the organizer to give me this opportunity to present my work here. And my name is Shengbao Suo from Goten Yuan's lab, and today my topic is mainly about revealing the critical regulators of cell identity in the multi-cell atlas. We know that for the single cell techniques have been widely used in different fields. And by using the single cell transcriptome profiles, we can easily identify different cell types. And with the rapid development of the technique or the improvement of different single cell protocols, and the number of single cells studied in different researches have been undergoing exponential increase in different in the past 10 years. We can see from this picture, and until now, multiple groups have been made tremendous progress in mapping cell atlas. For example, in complex organs and in earlier embryos, or even in the entire animals. And based on the international collaboration, the human cell atlas is on the way, and until now, it has already released about 300,000 bone marrow single cells from human. And from here, we can see that for the single cells, it is now opening a new way to study the cell heterogeneity. But the question is that whether we have lowered the cell composition of different tissues or even the hair species. But for most cell types, the regulatory network underlying their identity remains poorly understood. So mapping the gene regulatory network from single cells provide an attractive opportunity to study the cell heterogeneity. And the gene regulatory network is a set of genes that interact with each other to control cell type-specific transcriptome states, and which in turn underline cellular function. So there are different types of gene regulatory network. For example, the direct network and indirect network. And here we mainly focus on the TF networks, which contains active TF and the target genes. So this is the steps of identify the TF and target network. And we know that for the human cell atlas, it provides a very comprehensive annotation of different cell types in mouse. And it also provides a very good resource to study the regulatory network in different cell types. And in this atlas, it connects over 40 mouse organs and tissues and sequenced more than 400,000 single cells. So this is a global picture of the main cell types identified in the mouse from mouse cell atlas. So with this big data set, we built this pipeline to study the cell type gene regulatory network. So because we know that for the single cells, it contains many dropouts. So first of all, we use the aggregation method to improve the prediction, and then using the CELIC to predict the gene regulatory TF networks. And in this step, it contains three small steps. The first is to identify co-expression modules from the single cell expression matrix, and then the cis-regulatory motif analysis is used to identify the positive direct binding targets. So after that, we can get a set of regulatory networks. So here these are defined as the Regulons. So for each Regulon is a transcription factor, along with a set of co-expressed and motif significantly enriched target genes. So after that, we get a set of Regulons. So after that, we calculate the activity score for each Regulon in each cell. So after that, we can get a Regulon by cell matrix, and then we're using this new matrix to perform the analysis. So this is the results based on the Regulon activity score, and it is visualized by the t-SNE plot. It is a nonlinear dimension reduction method for visualizing the big data sets. So from this picture, we can see that for most of the cell types, it can be separated very well. And after that, we also focused on one tissue which contains different cell types. We can see that for the types contained in this tissue, we can see that different cell types can also be separated. So after that, we focused on the one cell type which is discovered in different tissues, and we can also see all of the stroma cells could be grouped together in this region. And what's more, we can see that we can still find, if you close look at it, you will find that the cells from different tissue also show a slight different based on the regular activity score. So the next particular question is that we need to predict the essential regulators for different cell types. So here we using this method, we defined a regular specificity score for each cell types based on the Jensen-Shannon divergence. So this is a matrix, the Regulon by cell matrix, and each column is a cell type. So for each cell type, we can calculate the Regulon specificity score. So for all Regulons, we can perform this analysis, and finally we can rank all of the Regulons like this. So the higher the specificity score is, the higher probability that this Regulon relates to this cell type. So the next we need to validate our prediction. So first, we use a well-studied cell types, that is the erythroblast, because we know the key regulators contained in this cell type. And based on this, based on our prediction, we say that the LMO2, GAT1, and TEL1 shows very high specificity score in this cell type, and which is very consistent with our understanding about this cell type. And the t-SNE plot provides additional support that those identified Regulons are highly specific to this cell type. So because we know that LM2 is the key regulators for the erythroblast, but for most of cell types, we didn't know this information. So we used another two methods to validate it. The first is we use these two methods to test whether to validate our prediction. So the first is the seq analysis. This is geo database mining method. It tests over 2,000 geo datasets with the assumption that the functional related genes should be co-expressed in the corresponding cell type. And the second is a coset analysis. It is a PubMed literature mining method, and with the assumption that the functional related genes and the terms or the cell types should be frequently appear together in the literature. So by using to validate these two approach are useful validation method. We tested the LMO2 Regulon, and from the result we can see that the Regulons the genes contained in this Regulon is very significantly enriched in this cell type. So based on this, we performed this analysis for all the other cell types, and this is another example of oligodendrocyte. We see that the THRA, this Regulon, shows very high specificity score in this cell type, and we also use the seq analysis and the coseta, and also confirmed that the genes contained in this Regulon is significantly enriched in this cell type. So next, we'll come back to the Regulon. So we know that for the transcription, functional related transcription factors, they mainly work together to regulate the gene expression. So here we just want to ask whether the Regulons also organize different modules to regulate different cell types. So based on the Regulon similarity across different cell type, we get this eight Regulon modules, and we also carefully checked the components of different modules, and we list representative transcription factors and their common bonding motifs and the enriched activated cell types. We say that for the group 1, for example, it contains GAT1, TAL1, and M2, and as I just mentioned, that it is related to erythroblast. And for the module 7, it is the biggest module. It contains the MAF family and the IR family genes. And it is mainly activated in different immune-related cell types. So here we ask another question about, so are there some sub-modules that contained in M7 that can reflect the hierarchical organization of the Regulon? So to answer this question, we're using the similar strategies. So we analyze the Regulon contained in this module, and we get these 5 submodules. And it is interesting that for these 5 submodules, we can see that this shows very high specificity activation in a part of immune-related cell types. So this analysis indicates a hierarchical organization of regulatory modules that work together in fine-tuning the cell states. So because we know that for the whole multi-cell atlas, it contains more than 800 cell types, so we applied the identified Regulons to the whole MCA dataset and built this cell type network. And by using the graphic clustering, we found that for the functional related cell types, they can group together, and we can visualize. The results can indicate by this word cloud plot. So, for example, for the group 1, we can see that they mostly contains different immune-related cell types, and this result also can indicate that for this for Regulon in this cell types, for cell types contained in the group 1, they maybe show some common regulators, but these regulators are totally different from other cell groups. So finally, to make this resource easy to be used by different biologists, we built a website for querying the different cell types and the different Regulons. And we also add different functions in the future and keep this website update. This is the interface of this website. We can roam in the big network and click the interesting cell types, and we can get the Regulons, and also we can get the correlated cell types. And this is a small video of visualizing this website, and it is very easy to use. So finally, this is a conclusion. So in this study, we computationally reconstruct gene regulatory network for different cell types in mouse. And we found that the cell type-related Regulons are organized into different modules. And we also predict a small set of essential regulators for each cell types. And finally, we built a website to query the multi-cell type network. So finally, I'd like to take this opportunity to thank my supervisor, Guo Senyuan, for his discussion and advice, and also thanks Chen and CR for the help in this project. And also thank my collaborator, Guo Guoji from Zhejiang University, and Li Jianfen. Thanks for your attention.
Thank you. We have time for one question, if there is any.
Thank you very much for the nice talk. Can you elaborate a little bit more about the network modularization part in your talk?
You mean this part?
Sub-module identification, basically. I assume it's some kind of hierarchical clustering.
Clustering?
The module, the regular modules.
You mean
Yes.
this one?
Right.
Okay.
Right.
Oh, you mean so this is just because we can get the regulon for all the cell types, right? So we can calculate the regulon similarity based on across different cell types. So we get this similarity score and perform the clustering to see whether there are some regulons shows very similar activation in different cell types. So this is just a clustering result that for one regulon for regulons contains in the one modules, they shows very similar activity score across some cell type.
Right.
Yeah.
Thank you very much.
Thank you.